Thank you for joining our presentation. Today, Kaz from Yamaha Motor and Ray from PhenoVista Biosciences present application of cell picking and high-content imaging in 3D models of disease. This presentation is divided into three parts. First, Yamaha Motor presents the cell picking system cell handler. Second, PhenoVista presents high-content imaging assay model and how the cell handler improves assay workflow. Finally, we have a live Q&A. Let's move on to the first part from Yamaha Motor. Thank you for joining our presentation. I am Kazunori Kochiya from Yamaha Motor. Speaking of Yamaha, you may think of motorcycle or piano company, but today I will introduce our new product, Cell Handler, which is an automated and rapid spheroid, organoid, and single-cell sort system. Let me start with Yamaha Motor. Yamaha Motor started our business in 1955. This slide shows our history and products. Among these mobility-related products, the Cell Handler was launched in 2017 in Japan. Why Yamaha developed biomedical instruments? The reason is we are actually strong in industrial robotics field, such as surface mounter. This is one of the examples of the surface mounter. As you can see in this press release, Yamaha launched the surface mounter with the super mounting speed in the world. You may have seen this type of green circuit board, but you may not know how it is assembled. Surface mounters are frequently used for the assembly of electronic chip modules for cell phones, PCs, and so on. The principle of chip mounting is shown here. Electronic parts are supplied on the rear. They are picked up by absorption, and the shape and direction of the parts are precisely and instantly recognized by a camera. These parts are then transferred and mounted on a board in a very precise manner. Actually, Yamaha is the world market leader in this category. This video shows the surface mounter in action. Electronic parts are supplied here and recognized here and transferred on this board. This is the actual speed. CPH stands for chip per hour, so this shows that this robot can transfer tiny parts very accurately at an ultra-high speed. Cell Handler was developed based on this technology. This is the Cell Handler. In this case, not electronic parts, but cells are transferred to not circuit board, but microtiter plate. The microscope recognizes spheroids, organoids, or single cells on a dish, and pipette tips aspirate and transfer them to another microtiter plate. To ensure accurate selection and transfer, we developed a special chamber and special aspiration tip. By using these, we can select transfer cells and collect data in a very accurate and high throughput manner. At first, let me ask you one question. If your cells are spheroids in the medium or organoids in matrigel, how do you sort them to 384 well plate? For the subsequent studies like drug assay or omics analysis, many of existing cell sorters cannot sort the 3D cultured cells, like spheroid and organoid, or you may not have sufficient cells, or the cells might be damage sensitive. Then, you may think dispensing by hand is the best way. But even if you try to dispense them by pipetters as one spheroid per well by limiting dilution, it will follow a Poisson distribution. So, as you can see, many of wells are empty or contain multiple spheroids, but cell handler enables to dispense single spheroid in each wells within 30 minutes for 384 wells, including image data acquisition. Next, I will show you how it works as a spheroid sorter. At first, spheroids or organoids are poured into the chamber, which has microcavity at the bottom. Images of the spheroids in the chamber are taken and immediately shown on the monitor, and the distribution of parameters are shown as histograms. In this case, particular size of spheroids are defined. Using these histograms, particular range of parameters up to 6 different parameters can be selected, like size, shape, and color of the spheroids. The cell handler uses special tip called pression tip, which is very thin plastic tip. To ensure the accurate picking, the position of the edge of the tips are calibrated like this. After the calibration, selected cells are picked from the chamber. In this case, pression chamber has microcavities at the bottom, and cells are placed in sections. The selected cells are accurately aspirated by pression tip and moved to a destination plate. 8 cells are dispensed simultaneously, and images are captured within 4 minutes, even if 384 well plate is used. After the transfer, various parameters are obtained again, like size, shape, and so on. In the previous slide, I showed automatic selection using histogram. The cell handler can recognize cells, spheroids, and organoids automatically and generate data for 20 parameters. And users can select up to 6 parameters out of the 20 and set the range from the histogram. And then the cell handler pick and transfer the cells that satisfy all the criteria. In addition, users can select target spheroids and organoids from images manually based on their preference from their own experience. After several iterations, the cell handler can learn the user's preference through machine learning function, and it can support the selection of cells. Here, let me explain how particular cells can be selected by the cell handler. This example was provided from Dr. Shimiz, who investigates cardiomyocyte from iPSC. In this approach, iPS cells are cultured in the spinotype flask. And as you can observe, the culture media contains many types of the spheroids. These spheroids were dispensed in the pression chamber, and images of cells in the microcavity were taken and shown on the screen. In this case, a user selected spheroids of interest manually. Using the cell handler, various types of spheroids were intentionally selected. In this case, pick and transfer success rate was more than 95%. After the 3 days culture, some of them were confirmed to be beating, thereby mimicking the heart function. So, we may be able to speculate what types of parameter of spheroids correlate with the proper differentiation. Therefore, the cell handler could be used not only for high-throughput cellular screening using spheroids, but also for quality management of cell product like iPSC. This is an example of spheroid selection by size. In this case, embryoid body of iPS cells were placed on the pression chamber and divided into 3 groups: small, medium, and large. And gene expression level were investigated. According to these results, undifferentiated state seemed to be maintained in smaller size group. This slide summarizes application of the cell handler. Stem cell research, cancer research, and single cell research are the focus area of our application. The cell handler can pick cells from various types of culture plate. One of the most unique functions is the direct picking of organoids from gels. In the next couple of slides, I show this unique function. As you know, usually organoids are created in dome-type gel, like Matter gel. This movie shows how the cell handler can sort organoids. At first, culture plate was set here and start to take images for the detection of the organoids. As you can see, organoids are immediately recognized automatically, and users can narrow down the target organoids from the histogram. As I said before, up to 6 parameters can be selected from the pull-down menu, and set the range of each parameter easily. And user can immediately start the organoid picking from gel. It means organoids can keep intact condition, and then the organoids are dispensed into destination plate for subsequent assay of individual organoid. Actually, this data was provided by Dr. Morimoto from Riken. He used several morphological parameters to define the organoids of interest. Not only such image analysis, but also the special tip, called pression tip, enables to pick target organoid directly from gel. Pression tip is a plunger-integrated type of disposable tip, and it can efficiently reach to the target organoid and aspirate it, as shown here. As you can see here, the cell handler successfully picked the target organoid, even in a very crowded position. And then, pression tip dispensed the organoid to U-bottom plate. According to Dr. Morimoto's comment, continuous growth and survival were observed, thereby confirming the damage-less sorting and transfer. We are collaborating with HUB, Hubrecht Organoid Technology, from 2019. These images were provided by HUB. You can see there are various types of organoids formed in matter gel, and only specified colon organoid has been successfully transferred by the cell handler. Under this collaboration, we are investigating the feasibility of precision medicine. Next two examples are cancer research using microcavity plates. In the case of collaboration with Dr. Inoue, the data for compound screening has been published. By using cell handler, cancer tissue-originated spheroids were dispensed into 384 well plate. As you can see, the size and appearance of the tumor spheroids are similar at T0. Outliers had been excluded, and Dr. Inoue conducted high-throughput compound screening using 2,500 compounds and identified several effective drugs. This example demonstrated that cell handler enabled to conduct high-throughput assay even using primary tumor. This slide shows the cell handler contributes not only to high-throughput screening, but also to assay accuracy improvement. This data was provided by our customer, Fukushima Medical University. As you can see here, they are establishing their own patient-derived tumor organoids called FPDO, and using them in drug sensitivity assays for pharmaceutical companies. In this case, the multiple numbers of PDOs were transferred to a microplate using the cell handler in order to optimize assay conditions and evaluate several drugs. This table shows the comparison of the method of cell transfer, manual, and the cell handler. 10 PDOs per well in this case. As you can see, the CV value and Z primary factor were improved dramatically by using the cell handler. Thus, These data demonstrated that the cell handler could improve not only the throughput, but also data quality. Next, let me show you another examples in single-cell research area. Not only the 3D cells, but also even single cells can be sorted by cell handler. This slide shows the example of single-cell clone selection after transfection. In this case, CHO cells were transfected with GFP gene and briefly selected by puromycin. And then single cells were dispensed in the L-pressure plate, and single cells with a strong fluorescence can be selected by histogram selection and transferred to a 384 well plate. 6 days after the cultivation, a formation of the single colony was observed. The cell handler can create tiled images to cover the entire bottom of the well. So, the cell handler will support the cell line development and confirmation of the monoclonality with high traceability. To ensure the accurate selection of cells, the cell handler has 4x and 10x magnification and 3 fluorescent filters. Next, let me show you an example of genome editing by single-cell selection. This data was provided by our collaborator HUB, Hubrecht Organoid Technology, as an establishment of clonal organoid after CRISPR-Cas9 genome editing. At first, organoids were disrupted into single cells and electroporated to introduce pCas9 GFP construct encoding the pCas9 nuclease plus sgRNA targeting the AAVS1 locus. Cells were cultured for 2 days and seeded onto a microcavity plate. The cell handler can automatically select single cells with GFP signal and transfer them to a 384 well plate. This table summarizes the results. Maybe due to some damages from electroporation, half of the cells could be expanded to clonal organoids like this. And 2/3 of clones were confirmed to have desired mutation. So, the process shown today enables the generation of CRISPR clones without any requirement of growth selection, even for experiments with low transfection rates or low Cas9 modification rates. And it would be possible to determine whether or not the target modifications are lethal. The next application is organ-on-a-chip. As you know, organ-on-a-chip has multiple small wells that may contain microorgan. In the next slide, I will show you how such cells are placed in appropriate position of the chip. This slide shows an example of application for organ-on-a-chip. Mimetas recently developed a novel organ-on-a-chip called organoplate graft that has 1 mm hole with a particular pattern of wells. In this case, spheroids were made in 384 well plate and transferred by the cell handler to appropriate positions of the organoplate. Since the cell handler can set dispensing positions precisely, it is possible to place the cells even in such a limited size and position accurately. Therefore, the cell handler could accelerate filling organoids in specified positions and preparation of ready-to-use organ-on-a-chip. And although I didn't show detail today, the cell handler can pick specific organoid from a particular well of the organ-on-a-chip to deassemble the system for subsequent individual analysis. In the next couple of slides, I will present extra unique functions and applications of the cell handler for 3D imaging analysis and 2D single colony picking. In this slide, we propose a new application for 3D image analysis using the cell handler. In the conventional method, for 3D image analysis, the wells containing spheroids are washed iteratively with PBS and change it to a fixation reagent or clearing reagent by manual operation. During this process, sample loss and spheroids collapse have been frequently observed. But in the case of the cell handler, it transfers spheroids themselves with a very tiny volume of buffer, like 0.1 microliter. Therefore, the washing process can be eliminated, and the risks of cell damage, sample loss, and contamination can be reduced. Furthermore, high traceability is ensured by taking images of the spheroids with various parameters before and after the cell transfer. This application will support robust data analysis and new discovery in 3D cells. Next, I will show you an example of this application. In this experiment, we evaluated the effects of a drug to a microenvironment of tumor spheroids using the cell handler and 3D cell analysis software Novisite from Olympus. Colorectal cancer cell lines, HT29, which is a sensitive cell line to an anti-cancer drug paclitaxel, and HCT15, which is resistant to the drug, were used. The concentration of paclitaxel was 2 nanomolar, the maximum concentration that maintains spheroid formation. First, spheroids with a diameter of 100 micrometer were created in a U-bottom plate and treated with paclitaxel at 2 nanomolar or DMSO as a control. After 3-day culture, spheroids were stained with ReadyProbe's cell viability imaging kit and transferred to a plate with a fixing solution and then treated with a clearing solution. As you can see in these thumbnails, which were taken by the cell handler, size and shape of spheroids were maintained during these processes without any sample loss. As expected, there was no difference in the area value between the two cell lines with and without PTX. However, the readout of fluorescent signal indicated PTX significantly reduced the proportion of living cells only in HT29 cells. All of these data were measured by the cell handler. Next, in order to analyze the effect of the drug to microenvironment of the tumor spheroids, we conducted image analysis using Novisite software from Olympus. As you can see here, paclitaxel reduced cell viability in HT29 throughout the spheroid, from outside to inside, whereas no effect in HCT15. In summary, the use of the cell handler in imaging analysis will improve the accuracy of 3D cell experiments by reducing the risk of cell damage, sample loss, and contamination with reliable traceability. In addition to 3D cell picking, the cell handler can directly pick 2D single colony from a culture plate based on morphological character. In this case, HT29, which is colorectal cancer, and H1975, which is lung cancer, were cultured in the same plate intentionally. As you can see here, HT29 and H1975 cells attached on the bottom as single colonies and shown round shape and spindle type shape, respectively. By using the cell handler, the colonies were picked directly and transferred to different wells based on the morphological traits, and clonal growth was observed after 7-day culture. Different cancer cell lines were used in this case, but during the process of stem cell differentiation or transfection, you will find various types of single colonies. Once you detach the cells, for example by using trypsin, then the morphology might not be maintained and it is hard to sort them without staining. But some staining of particular markers might damage the cells. By using the cell handler in 2D colony picking, enzyme processes are eliminated and The colonies were selected and sorted without any labels. As a result, the colonies are isolated with low damage and keeping intact condition. To sum up, the cell handler is an automated image-based cell sort system. It can transfer various types of cells, single cell, 2D colony, and 3D cells, with particular parameter from any position to any position in damage-free manner. That's it for now. Thank you so much for your kind attention. Next, I take over to Ray from PhenoVista Biosciences. He will present very interesting image analysis. Let's enjoy.
Hello, my name is Ray Price. I'm Vice President of Business Development at PhenoVista Biosciences, and I appreciate the chance to speak to you today about work that we've done in building both 2D and 3D cell-based models or assays of disease, and particularly how we've used Yamaha Motors and other partners to be able to build these assays and run them in a high-content screening format. Briefly, we're a San Diego-based biotech company, CRO. All we run is cell-based assay services. I'll talk more about that in just a minute. But in terms of disclosure, I don't have anything to say except that I am a full-time employee here at PhenoVista, but no other potential or actual conflicts of interest in relation to this. Really briefly, this is what we do for our day jobs, if you will. Essentially, we run high-content imaging assays across a range of different cell-based assays. So this typically starts from the preparation and obtaining of biological samples. So this can be iPS-derived cells, primary cells. Oftentimes, we're putting these together in co-culture systems. But doing something on the cell biology side, right, building and preparing cell-based assays, then running those through drug treatments, fix, stain, and image, and then really this reduction of images to an analysis piece, and then output features that allow clients and customers to make decisions about their programs, and really reducing this image analysis to a quantitative output. That's really key. We're quite flexible in terms of how we work, so we'll take inputs. These are often built on a kind of core assay concept where, for example, taking neurons from a particular cell type, we might not have worked with that particular assay before, but certainly can apply features or assay components, like I'll show in just a minute, around neurite outgrowth or synapse formation, and apply those to a novel cell type, maybe at different time points or in different co-culture environments. That can then move into a development phase where maybe we optimize assay windows and then move into screening. We've also built assays, on feasibility and development, and transferred those back out to clients. And we brought in assays. So figures from a paper, right, can often form the foundation of a particular assay. Or a client may have an assay that they have set up and run on a confocal microscope, let's say, but want to miniaturize and automate further. In which case we can bring it in and then move into a screening. And screening here doesn't necessarily mean only sort of the traditional high throughput sense, but could be, looking at maybe a small number of compounds, but just, really deeply interrogating the cell biology that's present there. Example of these assays is shown here, where we're taking immuno-oncology related compounds and looking at their ability to potentiate the immune response to tumor cell death. So essentially creating a co-culture of immune cells with tumor cells, and then looking at how compounds improve tumor cell death. Here, if you're not used to looking at imaging readouts, the first column, the images are generally organized in columns. So HUX is a nuclear stain. Here we've labeled the tumor cells with cell tracker green dye, so it's taken up as fluorescent and viable cells. Similarly, for PBMCs, a cell tracker red dye, and then DRAC7, which is a marker for dead cells, and then a composite that combines all of those components. The first row is just the vehicle, and then the second is a compound that is known to or was thought to be involved in or important in this immune-related tumor cell death. And in fact, we can see that. So certainly there's a decrease in tumor cell number compared to the DMSO control. But interestingly, and it's kind of obvious, or maybe more obvious in this composite image, we can actually see this induction of immune cell clustering. Right? So These immune cells are for some reason or somehow induced to cluster around the tumor cells, and that can, if we measure that over time, we can certainly look to see, does that precede or is it required for tumor cell killing or not? And we can do this in a really in a screening type format. So essentially, the idea here is to look at that across a range of different compound types, right, using 2 different doses. And we actually, not shown here, we did this with a range of different stimuli. We could even do time points or cell types, right? But the top graph here just shows a viable target count. So we're able to essentially measure the tumor cell death specifically. We use a statistical analysis called SSMD scores, strictly standardized mean difference. So essentially, this is the number, it takes into account both the difference in the mean as well as the difference in standard deviations between 2 data points. And you can see a number of compounds were significantly able to reduce tumor cell number in this co-culture assay. And we also saw that many of them, but not all, there were certainly some compounds that did induce tumor cell death, but that didn't increase immune cell clustering. And from a matrix of these features, tumor cell clearance, T cell, both activation and proliferation and clustering were able to really generate but distinguish these cell populations from each other. So certainly the argument can be made that we don't live our life in dishes or on plates, and the ability to convert some of these assays to a 3D environment certainly could improve the physiological relevance of the assay, and particularly if we think about doing that not just with a more relevant sort of 3D structure or format or biology, but really combining that with primary tumor assays. And the way that one way anyway, that we've chosen to do that is through patient-derived xenograft or PDX models. This work was done in partnership with Champions Oncology. They have an extensive collection of more than 1,200 different PDX models across diverse tumor types, and on their end have really extensive both clinical and molecular characterization. So the model here, or the way that these tumor systems are developed, is to take a primary tumor from a patient, engraft it into mice and passage it and grow them up and be able to essentially carry the tumor component that from the original tumor in the mouse, and then be able to, in our case, then dissociate those PDX tumors. So what we get is essentially a cryopreserved chunk of that tumor. So we dissociate those, create these tumor fragments, and then culture those into a 384 well plate. And then, can combine that both with high content imaging, sort of what we do traditionally, but also other viability dyes and other ways to measure cell viability. And, this is an example of what those fragments look like before sorting. So, again, the idea is to create tumor fragments from a whole tumor, essentially. And we have also run these assays with primary tumors, with patient-derived organoids and other primary cell types as well. But certainly the idea is to be able to create a platform that allows clients to be able to access sensitivity and resistance assays using a complex primary tumor-derived material. And again, a large part of this ability to work in 3D, if you think about taking this heterogeneous, clearly heterogeneous tumor population with various fragments and, from single cells up to really large tumor fragments. One way to optimize that or to improve the assay window is to be able to sort these reliably and with a variety of different selection criteria. And so this cell handler can actually do those things. So it is built to, be able to, in the middle here, as we heard in the last section, right, the ability to place, either in plates or in special chambers, a large number of these tumor fragments, and then be able to use in the selection on either with bright field or fluorescence-based selection processing, move those into a 384 well plate, and precisely be able to select tumors that are 200 microns, that are perfectly round, that have a certain optical density, and therefore could be healthier. And so this alliance with both the Yamaha Motors as well as Champions has really enabled us to build out a 3D patient-derived tumor model platform. And again, this cell handler is certainly able to do different things besides just pick from media. The idea here is to be able to identify and optimize a set of cell models that are optimal for screening. So here again, if these are gut organoids, the ability to select for and even from a hydrogel grown, patient-derived organoid platform, be able to start off with a really consistent, uniform starting material that really allows you to have your assay be as robust as possible. And this selection can be image-based, or it is image-based, and can be guided by features like size or shape, fluorescence, optical density, etc. So here we're looking at these PDX-derived tumor fragments and then treatments with various small molecules or combinations. So again, to orient you, the dyes here are shown in rows. So Hux again is a nuclear dye. Cell Tracker is again a viability dye, so it's taken up and fluorescent in cells that are viable. And DRAC7 is a dead cell marker, so we're labeling here dead or dying cells. And then a composite image, and you can see a pretty clear difference. Here, for example, in the control wells, right? A lot of good nuclear staining, viable cells are quite the number of viable cells and large fragments that are present, and fewer dead cells certainly than at least these other tumor fragment components. And then with various drug treatments, either drugs alone or in combination, right, we can see almost complete reduction, for example, in the Cell Tracker dye with these two treatments, and certainly much smaller fragments that are surviving. A lot more DRAC7 positive cells, indicating a lot higher rate of cell death, even if there are some Cell Tracker green positive cells here. And again, the idea is to be able to quantitate these features, so taking the advantages or the benefits of a high content system, right, is being able to measure things like metabolic activity here, the Cell Tracker red dye in this case, and being able to normalize that across wells and generate dose response curves in response to a particular compound treatment. We can also incorporate, through these high content systems, the ability to measure other readouts. So here we're looking at a readout called EDU incorporation. This is a marker of proliferation, and more specifically of S phase cells that are present in, or that are just in the S phase of cell division. So Hux again, nuclear stain, and then the EDU, so only two dyes here, and then between the negative and positive control, right? So a normal level of cell proliferation, and very little, if any, detected in the positive control drug. So this is a drug that should block almost 100% of the proliferation. And then a chemotherapy called osimertinib that is two different doses, either completely stops it or is certainly reduced compared to the negative control treatments here, and that's quantified here. So again, these are SSMD scores. What we're looking at are the difference, in terms of number of standard deviations between two different data points, and then looking at two concentrations of a range of different drugs and how that each of these drug treatments reduces the EDU incorporation, or the percent of cells that are in S phase. And similarly with phosphohistone H3, this is a commonly used marker for mitosis, or cells that are undergoing the mitotic stage of cell division, and using an antibody specific for phosphoserine 10 on histone H3. This is tightly correlated with chromosome condensation, and we can use this as a measure of cells, the percentage of cells or the number of cells that are present in this stage of mitosis. So switching gears here a little bit, we'll move now into some of our work in neuroscience. So this slide lists a range of different neuronal health or CNS or neuroscience related assays that we've developed. And as we mentioned earlier, the idea here is to be able to apply these to a range of different either iPS-derived neurons or primary cultures. We've done a lot of work now with co-culture of neurons and astrocytes, as well as more recently some neuron astrocyte and microglia triculture work that we'd be happy to talk about. And these features, can be applied again to a range of different um, neuronal types. So we can take, cells from Parkinson's patients or Alzheimer's or ALS patients and measure, many of these features. Right, compare sort of normal wild-type neurons, if you will, to an isogenic comparator neuron that might have a particular mutation. That's important for how that disease manifests either by genetic association or by mild stresses like NPP plus or other compounds that are known to impact those particular neurons or be neurotoxic. So these features can include things like neurite outgrowth, ER, mitochondrial stress, autophagy, synapse formation. We've measured features like glutamate toxicity, either in the presence or absence of astrocyte culture. Again, this idea of neuron microglia interactions, being able to measure synaptic pruning and features like that. We've looked at neuromuscular junctions, and then certainly have put these into other 3D structures or microfluidics cultures, blood-brain barrier and other models like that. One example of that is shown here. This is an example of neuronal network analysis using iPS-derived neurons. So fairly common type of assay for us. Here looking more at toxicity as opposed to perhaps neuroprotection or neurotropic effects. And again, the two markers here are HUX, the nuclear stain, and TUJ1, which labels neurites, microtubule proteins in the neurons. And what we've done showing here is a composite image of those two features, and then nuclear mask that labels the nuclei, allows us to generate and quantify features that rely on, for example, total number of neurons or total number of viable neurons. And then a neurite mask, where the software is automatically identifying those neurites against the black background here. And again, it's not just simply the ability to collect these images or to really be able to visualize what's going on, but also to be able to quantitate these. So, again, DMSO control feature set that gives a baseline level of neurite network or neurite outgrowth. We can block that or disrupt it, with small molecules. We've done siRNA testing, again, genetic models of disease, as well as molecules that are known to potentiate neurite outgrowth, things like psychedelics or other epigenetic modulators that are known to, in the literature, improve neurite outgrowth and have neurotropic effects in these systems. But really being able to quantify these features. So your neurite area per cell as a kind of arbitrary unit, and being able to see that decrease with this compound treatment or increase, right? And be able to measure our dynamic response in both directions. And this is done, in 384 well format. We can normally or typically test hundreds of 384 well plates a month. So really the ability to do this, either on a small scale screen, be able to look at things like combination studies or different time points or features, or fairly large screens that we can prosecute. On the kind of other end of the spectrum is this synapse formation assay. So here, again, the same neurite marker that we've used before, TUJ1. The middle is a presynaptic marker. Often this is synapsin or synaptophysin. And then a postsynaptic marker like PSD95. And here we've told the software to basically identify in each of these three feature sets, where is there a coincident detection, or basically where are these three signals overlapping or occurring within a certain minimum distance from each other, right? And then being able to find those co-localization events through all three sets of images. And again, reducing these to a quantitative readout. This really is key to what we do.
We can take a similar approach here to be able to take these 2D neuronal models that we use quite a bit to move that into a 3D environment. And here doing that with microbrains from a partner called Stemonics. In this case, this is a mixture of GABA and glutamate neurons combined with astrocytes, particularly glial cells. And here we have the ability then in a really uniform set of starting material to be able to measure things like neurotoxicity, right? Cell-cell interactions. Are you selectively improving or disrupting particular neuronal types, for example, and doing that in a really robust and reproducible 3D environment. A similar approach and kind of classic phenotypic profiling is shown here. One example is a technique called cell painting. Essentially, here what we do is take a range of different dyes that label different cell compartments, and then use that to inform, essentially create a phenotypic signature for these either compound responses or siRNA or gene therapy or various different perturbations that can impact the cells using a range of different dyes that label different cell compartments. So the idea is really sort of essentially what phenotypic screening is, right? Using the same set of dyes to be able to unambiguously, but on a non-target based approach, identify and target different cell compartments, and then use that to create a set of hits or feature sets that are the most informative, and then use techniques like machine learning and principal components analysis to really interrogate in that database of responses which reference compounds or compounds that are known or siRNAs that impact a particular target. How do they change, the cell phenotypes, and then using that to be able to deconvolute how another set of compounds or features is working.
Now, similar to the other two examples, we're going to take this 2D application of cell painting and apply it to a 3D, structural environment. So here, these are showing gut organoids that are from a company called Definigen, one of our partners. These are grown in a hydrogel format or environment that allows them to create illuminated structure. We'll see that more in just a minute. But again, the idea of being able to use the cell handler to, from a range of different cell systems, right? Some of these are clear and round and not very healthy or very differentiated, whereas some of these are lumpy and dark and very healthy and on their way, let's say, to creating the kind of organoids that we would ultimately be interested in terms of the features and illuminated structures and the 3D structure that's ultimately quite interesting. And that's shown in more detail here. So just the inset here is, we're going to zoom in. And you can see that these guys are quite lumpy. There is maybe hard to see a little in bright field, but there is illuminated structure that's forming inside of these. And so, because the cell handler can pick from a hydrogel, where we're able to tell it in this entire well where we've grown these structures, pick the ones that are more consistent size, shape, optical density, and then transfer those to another system to be able to grow further. And if we do that, then we can generate structures like this, right? So we're just looking at different Z stacks or Z planes above the plate. And this is, again, applying those same cell painter dyes that we talked about before. Nuclear, ER, WGA, and Floyd, this is cell structures, and then mitochondria for our mitochondria, for mitochondrial function. And you can see there is a illuminated structure in here. So essentially there's an epithelial cell lining. There are enterochromatin cells in here. There are other cell structures and cell types that are present as well. But, really for something like intestinal toxicity or microbiome studies or things like that, really, the ability to create a 3D structure, besides just in a trans well or something like that. Could be valuable depending on the assay and the research questions that a particular scientist has. And so again, how we've integrated this instrument into the workflow is really the ability to select, right, and image prior to and after transfer to confirm transfer to show if there is, it's very rare, but if there is disruption of the spheroids or organoids during the transfer process, and really being able to create a consistent time after time, assay plate for us to be able to run our kind of standard imaging assays on, and be able to, for example, qualify, fairly large numbers in this case of, markers, right? A process we call palette optimization. So the ability to select for and optimize your antibodies and dyes and combinations of those to be able to interrogate, particular aspects of disease biology, whether that's proliferation or synapses or features like that. But really the ability with this instrument to be able to create and allocate 3D structures, whether they're spheroids, organoids, PDX models, into a uniformly consistent micro well plate format for then subsequent staining, fixing, staining, imaging, and analysis. So in summary, then, we definitely, as a service company in this space, has seen the acceptance of more physiologically relevant models in the past few years. Particularly in our workflow, which is typically phenotypic assays, but certainly the advent of high quality iPS-derived neurons is one example of that. The growing acceptance of PDX models and other ways to model as close as possible to the primary tumor or to the diseased neuron, right? The ability to apply those and do that both in 2D and in 3D, although certainly one thing we haven't talked about on the 3D side is the analysis becomes more complicated. The imaging time is longer. Staining is not trivial all the time. Especially if you're looking at structures 150 or 200 microns and larger, the application of clearing or other methods to do that. But certainly, instruments like the cell handler as well as others, at least at PhenoVista, we're quite familiar with the challenges and the ability to both, run 3D cultures, drug treatments, uptake of, siRNAs or AAVs or other vectors, and then being able to image and analyze those on a really robust and high throughput fashion. I'll stop here and say thanks again to our partners, Yamaha Motors in particular, and sharing the session with them today, but other StemOnyx champions, oncology, Definigen, and then Fujifilm, our CDI, who provided a lot of the neurons that we talked about at the beginning. And happy to certainly hear from folks. Our website's here and my contact information. And thanks again to Yamaha Motors, to ISSCR, and to our partners here for allowing us to provide hopefully an interesting snapshot into how we work here at PhenoVista. Thanks very much.